And good morning, one and all. And how many of you from chemistry background? Who are like, yeah, two of them are from chemistry. And how many of you from biology? Okay, most of them from biology. And biochemistry? Oh, one person is from biochemistry. Good. So I will try to keep my lecture towards like a biology oriented approach, because most of the students are from biology, but I will give some flavor of the chemistry as well. So, how many of you know what's a glyco-hybrid? This is a bit new term. You know hybrid? What's hybrid? When you mix two things, hybridization, which we all study at 12th standard. The S block, S orbital and P orbital mixed together. So basically mixing of two together is called hybridization, isn't it? So glyco-hybrids term itself, self-explanatory. That glyco means sugar, carbohydrates, and so any molecule which are formed by the combination of sugar moiety, they are glyco-hybrids. So if this is specifically bonding at the anomeric position in carbohydrates, those are called glycosides. We all know that there are O glycosides, N glycosides. When there is sugar molecule, carbohydrate molecule, when they are attached to the anomeric position, there is glycosides. So glyco-hybrids means there could be three types. One is if there is, let's say, two molecules are joined together, okay, through some linker, that is called linked glyco-hybrids. If they are attached like this, so then you can say they are merged glycosides. If they are mixing like together, where the identity of glucose is not anymore there, then you can say merged, this overlapped glyco-hybrids. Okay, so fused, merged and linked. Basically the third one is the fused glyco-hybrids, because they lost their identity. Clear? So in today's lecture, in next 60 or 80 minutes, what I will be talking about the synthesis of these glyco-hybrids molecule, and basically where there is a natural product scaffold is there. So why natural product scaffolds? Because if you talk about the medicine, if you talk about the medicinal chemistry, so I am sure that your teacher has taught you that most of the molecule which is used as a drug candidates, a drug molecule, they are originated from nature. 70 to 80% drugs, they are either natural products or natural products derivatives. Okay. Natural products means just taking the molecule, let's say Taxol. Taxol is a natural product. You might have heard any other simple molecule you can think about. Curcumin. So, okay, curcumin is a natural product. But when you do some modification on it, that is called derivatives. For example, art ether. Artemisinin is a natural product which cure the cerebral malaria, and its methyl derivative or ethyl derivative, the methyl derivative was made by China, which enhances almost 10-fold activity of artemisinin. Artemisinin is a molecule, okay, and actually discovery of this molecule also given the Nobel Prize. Artemisinin, you might know about it. Its methyl derivative and its ethyl derivatives 100 times more active, and that was discovered by India. And there is one lab in Lucknow which is called CMAP. How many of you know CMAP? Central Institute of Aromatic Medicinal Plant. There is a laboratory, CSR laboratory. So they discovered they actually isolated the artemisinin, and then CDRI, Central Drug Research Institute, they have modified it. So means most of the molecule, what I am trying to convince you that 70 to 80% of the molecule, they are either natural products or natural products derivatives. Okay. Therefore, when we aim for making some biologically active molecule, or when we aim to work on the medicinal chemistry or in the pharmacy, we always rely on the natural product derivative. So what sort of glyco-hybrids I am going to present? They are all designed and made from the natural product scaffold, or you can say natural product inspired synthesis of glyco-hybrids. Clear? So This is very common slide, which you have seen several times in your studentship, that the drug discovery process or discovery of a new chemical entity or a drug molecule is not like a one-day job, it's not like a 1 month job, it's not like 1 year job, although this is 10 to 15 years. For discovering one drug, if you are getting success at each and every steps, it takes almost 10 to 15 years. And it includes a lot of finance, and the first step in the drug discovery process is, first is the target identification and target verification. Let's say if you are making the drugs, so what's target you are going to target it, and what's their structure, what is the structure of protein, enzymes, and what are their natural ligands, and how the molecule which you are designing, how they are going to bind to them. Okay. So very first step is the target identification. For example, if you think about in our daily life, let's say if you want to become, if you want to get a master degree or PhD degree, you have target few universities, that these are the universities where I want to go to get the PhD, and these are some professors to whom I want to work with. Okay. So this is very preliminary step. So target identification in drug discovery process is very crucial, very important step, and it takes not much time. It depends on like how quick you can do, therefore time is not mentioned here. Generally it can take 6 months, 3 months, or even 1 year. The next step is, once you got the target identification, you have to do the assay development. Let's say this is the target. So if you have got the molecule or you have made the molecule, so what's assay you are going to use to assess the biological activity or biological function of your molecule? Okay. So it could be enzyme based assay, it could be cellular based assay. Okay. So assay development. Then next step, which is very crucial one, here you make lot of molecule. The chemistry role comes here, and where you make the molecule like lead discovery. What is lead discovery? Is making large number of molecules and then screening them and finding few hit. Okay. Let's say you have made library of 1000 compound or 100 compounds, and you got 5 molecule as a active molecule, that is called hit. And then from those hit molecule, you do the lead discovery. From those 5-6 molecule, you have modified them, you have make them more potent molecule, and you got 3 molecule or 2 molecule as a lead molecule. Then from lead to you got the next step. So this step, third step, this is here chemistry comes in picture, high throughput screening, and in this one, and then once you got the molecule, then you do a lot of pharmacology, ADME and ADMET and efficacy. You know what is ADME?
Absorption, distribution, metabolism, excretion.
And T stand for toxicity. So this is also a very important study which has to be done in collaboration with the biologist and chemist, because if there's some toxicity is coming in your molecule, so what sort of group has to be removed, or what sort of group need to be installed to balance its toxicity. So this is ADME, and this all together takes almost 6 to 7 years. And what are the small component involved here? So first is hit generation, and like hit identification like a small molecule. Then from hit to get lead molecule, as I mentioned, let's say you have collection of 100 molecule or 1000 molecule, and you got 4 or 5 hit out of that, then you have developed into those lead generation, like few molecule as a lead molecule. Then you do the lead optimization, and then from lead optimization you may get 1 or 2 as a drug candidates, or the new drug molecule which which is suitable for the biological system. Then once you got the lead optimization and the one molecule which you think that you found that okay, this is the one which is drug which qualify all the parameters of the biology, then you go these 3 very important steps for the which is called clinical trials. Phase 1 clinical trial, phase 2 clinical trial, and phase 3 clinical trial. Okay, so what are the phase 1 clinical trial? In phase 1 clinical trial, you select almost, actually here in all 3 the fundamental difference is the number of volunteers. Okay. In phase 1 clinical trial, you take the volunteers, which is let's say 100 to 1000 in number, and also they can be from one local area. For example, if someone is doing the drug discovery and they found the lead molecule and they finally is a drug candidates, and then they selected let's say 100 people from NCR region. Okay. So, and then they did the study in the human trial and found, okay, this drug is working fine and there is no toxicity. Okay. Then once qualified the phase 1 clinical trial, then you have to go phase 2 clinical trial, which in fact the number of patients, these are healthy volunteers, this is number, and also the phase 1 is on the healthy volunteers. Phase 2 comes to the patient volunteers, which can vary up to 5000. So number got increases, and now let's say you are developing molecule for the anti-cancer, so you found a patient which are suffering from the cancer problems, and then you visited different hospitals and then taken the consent, and then you have done the phase 2 clinical trials. And the phase 3 is which is long term use, and where you look for their efficacy, safety, and the number of volunteers can vary up to 50,000. You see, this is the number, which is quite huge number. And these 50,000, they vary from 1,000 to 50,000. They could be 10, 15 as well, but they has to be from different geographical location. That is very important. Different geographical locations means you have taken few volunteers from let's say Delhi, few from Chennai, and few from let's say Dhaka, few from Islamabad. So there has to be, global kind of volunteers if you can find, internationally volunteers. That would be the best test for the phase 3 clinical trials. And this is the stage where most of the drug failed, because you have spent almost 10, 12 years, and finally you find, because here you are doing the study to the long term, let's say you have given the drug, you have observed the patient for 1 month, 2 months, 3 months, and you found that, okay, drug is working fine, but it's causing the liver toxicity. It's causing the accumulation of drug into kidney. So that means that drug is okay, it's curing the disease, but at the same time, it's killing the, let's say, bacteria, but at the same time killing the person as well. So it cannot be safe drug, isn't it? So most of the failures in drug discovery process comes into the last stage, which is phase 3 clinical trial. Is it clear? So it altogether takes 10 to 15 years. So like this is 7, and this is 8 to 10, and then for 14, and then 1 year here somewhere, 6 to 1 month. So 14 to 15 years. And if all phase 3 clinical trial also qualified, then that becomes FDA approved drug. Food and Drug Administration Department, and that department exists in all the countries. For example, if you want to sell your drug in Britain, so you must get the approval from FDA Britain, not the one which got approval from India, so that cannot be valid. Okay, is it clear? So now I hope I have given you some glimpse, like what are the steps involved into drug discovery process and how many steps are there. And you might aware about these slides, but I have just repeated it again. Next is why we need drug. Why? Because definitely we need drug to treat the patient. And why we need new drugs? Because the drug is already in the market. Because you think about any disease, drug is there. And not only one, you get 2 or 3 types of drug. For example, if somebody has fever, there is a paracetamol, Dolo, and there is another one. What is another molecule for an antipyretic? Azithromycin is not for the fever, it's for bacteria. There is yes, aspirin. Okay, aspirin, which is a product of salicylic acid. Okay. So then why we need drug? Because if already there in the market, so why we need then drug? We need a drug because as when some drug is getting used for a longer period of time, the bacteria or any particular disease or fungus, they develop resistance. They know how to escape from that particular drug candidates. So, and see, in order to overcome this problem, we need a new drug which must have the different mode of action, new mode of action. Because if the drug is acting in the same way, so that means bacteria know, for example, those of you from farmer background, you might be knowing there is a one which is called nilgai in the, which actually eat the, our crops. So over the period of time, farmers started knowing, okay, this nilgai or this particular animal is coming from this direction. So they try to fix those boundaries. इधर से नाप है. Similarly, bacteria, they also learn, okay, this molecule is act like this, and this comes and this act like this. So let's develop a resistant. So the new molecule which has to be developed, that must have the new mode of action, new way of killing the bacteria, new way of killing the fungus. Therefore, new drug is required. New drug is essentially required because the resistance is coming into the many bacterial infection and even the metabolic disorder disease as well. And there are two types of diseases. One is pathogenic, other is non-pathogenic. The one which is caused by pathogen, let's say bacteria or fungus or algae, and other one which is metabolic disorder like diabetes, like cancer. Only one type of cancer is infectious, other are metabolic diseases. So these diseases like cancer also becomes resistant. There are many anti-cancer drugs, and some of tumor, they become very resistant, solid tumor. So therefore we need a new drug, and these are some graphs which this is actually global position, global antibiotic resistant, and many bacteria, they have been found resistant for many antibacterial agents which are currently used. And in India also we have this Salmonella typhi, which is called typhoid, which is also very problematic disease, which is actually food-borne disease, or you can water-borne disease. So then we have superbug also in Delhi, and so this Salmonella typhi was found resistant in India in 1990s, and there are many. So not only India, almost all different countries, because not much information from China, but if you see in Europe, we have bacteria there also which is drug resistant, which was found in 1988. Then in France, almost same time. Then USA, okay. So USA is a developed country, still they have many bacterium which are resistant to antibiotics. Then South Africa, Australia, Philippines, Japan, Germany, and India. So that means if you see globally, there are many strains of bacterium, they are drug resistant. Okay, so finding the drug for them or finding alternative, either combination therapy or new molecule, is essentially required. So therefore, we need a new drug molecule. Okay. So next is, this is the chart which I have recently taken from the website, and where it shows how the expenditure for drug discovery got increases over a period of time. So if you think about, this is actually annual expenditure for which is in billion and also number in dollar. So like in 2001, it was almost $29.8 billion expenditure people are putting into the drug discovery, finding the new drug. As the new diseases started coming, as the resistance develops, this cost is keep increasing, and by 2021, that was the time, 20 and 21, the COVID time. Okay, you all must have experienced it. The cost gone up to $102 billion. And this is the expected data. This is the current one. Like in 2025, this is from 102 to 24 billion further increases. Now this is to 124. And then in 2026, which is the current year, this is like expected one. This could be around $142 billion. And then by 2030, this is almost $250.5 billion US dollar. Okay, so lot more money actually coming into the pharma industry for the drug discovery, for the making new molecule as the new problems are coming. So that means this particular area of research or discovering molecule is very much in demand and very much in contemporary. Now, what are the way, when you think about as a scientist, as a student, or as an academician, and this particular slide you might know a bit of it. Let's say when we want to go and I say, okay, I want to be a medicinal chemist. I want to discover a drug. So we should know background. Let's say if somebody wants to be a businessman or somebody wants to be a farmer, so they know in which particular season what crop has to be grown. If you want to be a businessman, we must know in which season what, depending on the business, what sort of business I should do, then I can be successful. So similarly, in medicine, if somebody wants to pursue their career in medicinal chemistry or drug discovery, they must know what are the way by which new molecule has been discovered, and what are the current scenario. So the slides on in front of you. The very first one, by chance discovery. Okay, so in drug discovery process, few molecules, they have been discovered by chance. And the best example is penicillin. You know history of this. I'm not going to tell you the history of how the penicillin was discovered, but I will ask when it was discovered. 1928, very good, so by Alexander Fleming. And there is a long story you can find out in the Google, and this was by chance discovery. And because for structure establishment, the University of Oxford was also involved in that. There's a inorganic chemistry laboratory, which actually I have visited because I have been in Oxford for quite a long time. So there is a professor who discovered the structure of this molecule. Okay, penicillin as an active molecule was discovered, but the structure was not known. The structure was known much later, like in 1930s. And when it was first FDA approved, when it was first approved by FDA, actually this was a drug which was used by the public before it was approved by FDA. This is also good to know. It was approved by FDA in 1945. And 1945 was the era of Second World War. What is the time for Second World War? 39 to 45. Okay, so August 45, when USA bombarded Nagasaki and Hiroshima by atom bomb. I think that was the time when the Second World War ended up. But before that, around 1939 to 1945, the war was happening around the globe, and many soldiers were getting injured and getting infected. And this was the molecule was used very much there without its approval FDA. ठीक है, इसका approval नहीं था, but because it was acting, so it was very much used throughout the globe. So, but FDA approval comes in 1945. Molecule was discovered in 1928. Long, almost 15, 16, how many years? 17 years.
So was it not causing hypersensitivity?
It was not causing
Hypersensitivity, because penicillin is known for causing all type of hypersensitivity.
Penicillin was actually before, during World War, when people are using it, they are using it as an ointment. Let's say some infection is there, some injury is there, and then some in ointment form was getting used, and it was actually curing the bacterial infection. Anyway, so this discovery was the by chance discovery. Second molecule which you see here, which is from there are many, but this is very example which I covered here. This is there is a one bincristin, binblastin. There is even salicylic acid is a natural product, and its derivative, if you acetylate the OH position, it becomes aspirin molecule. So this is Taxol. Taxol was the molecule. Taxol is the molecule which has been isolated and very effective for the cancer treatment, and this is one of the prescribed drug for many type of cancers in the even current days. So this is natural product. Then third approach was by doing the clinical observation. Okay, so let's say somebody is discovering drug for antibacterial, but when they are doing testing, they found, okay, this is also acting for some other purpose solver. So for such molecule is the Viagra. And then fourth one is the natural ligands. Okay, what is the difference between natural products and natural ligands? Natural products is like the one which has been isolated from the plant product. And natural ligands is also very similar, but these are one which, because they have protein, you have enzymes. And these proteins and enzymes, they have binding pockets. And in those binding pockets, there are some natural compounds, they are naturally fitting there. Okay, they are very much, very much occupy that binding space, and those called natural ligands based one. So then based on those structure of those natural ligands, you can think about making new molecule. And the third one, by searching the new molecule from the existing drug, which is called reverse pharmacology. You heard about this term, reverse pharmacology, or repurposing the drugs, okay, which was very much seen during the COVID time. There is one molecule, what is the name? Hydrochloroquine. This is anti-malarial molecule, but this was used as a maintaining the oxygen level into the COVID patient, because that time even the president of USA, he was also taking this because there was news. I don't know because I was not doctor after he was president, but there was a news that he was taking this particular molecule to not to get infected with the COVID, isn't it? So this is called repurposing of drugs. Like that particular drug is used for one purpose, and because they're all pharmacology, absorption, distribution, and those 15 years. Second slide, remember. So those 15 years you can shorten by a few years, because you know what is the property of this molecule. So can we repurpose it for this particular? It was used here. Can we repurpose for different use? So that is also called from the existing drug. These one, like last 10, 15 years approach, like one is high throughput screening, as I mentioned, making large collection of molecules without knowing anything, because you made the molecule and then maybe you are thinking, okay, out of these 1,000 molecule, maybe I will get 5 hits. There are chances you get, and there are also chances you may not get. Okay, and the next one is the diversity oriented synthesis. So what is diversity oriented synthesis? The protein molecule, they are 3D structures, they are having 3D structures. So that means their environment is very much exist in 3D space. I think now people are talking about 4D space, isn't it? 1D, 2D, 3D. In this AI era, people talk about 4D. So anyway, so 3D space is a three-dimensional structure. In those three-dimensional structures, what the diversity-oriented synthesis actually aimed for, and this concept was given by Stewart Shriver in the Harvard University. Okay, Stewart Shriver, S. Shriver. He is still alive, and he's I think more than 85 or 90, around 90-year-old, and still he is very active. And he has his own company. Anyway, so Stewart Shriver is given the diversity-oriented synthesis concept, and in this concept, what he proposed that it's not necessary to it's necessary to make large number of molecules, but those molecules should not be like a similar scaffold. They must occupy the diverse 3D chemical space. Okay, in high throughput screening, let's say you are making the molecule, you have one scaffold, core scaffold, and you are modifying, let's say you put the methyl group, then doesn't work, then you put the ethyl group, that doesn't work, then you put the propyl group, doesn't work, then you put the butyl group. Okay, so this is how you are making the large number of molecules. But three-dimensionally you are not changing much. So then chances of getting hit to lead are less. But what Shriver has proposed, and that make the molecule, large collection of molecule of course, but that has to be diversity. Three-dimensional diversity. There has to be stereochemical diversity, there has to be skeleton diversity, there has to be appendages diversity. Okay, so what are those diversity? I will come I will explain you what in coming slide. Stereochemical diversity, you can think about very simple example I would like to share. You know the structure of glucose? Just think about the structure of glucose, where the hydroxy exists, cyclic form, and galactose. What is the taste of glucose? Glucose is sweet, isn't it? And galactose? Anyone? Galactose is not sweet at all. And if you think about their molecular structure, exactly same. There is no difference. Anomeric is same.
And the carbon position, carbon number 4.
Yes, epimer. So the carbon 4 in galactose is axial, and in glucose this is equatorial. So this small change, this versus this, totally changed its biological function. One molecule is sweet, other molecule is not sweet at all. So this is called stereochemical diversity. In one case this is bond like this, another case bond is like this. So now this is occupying this 3D chemical space, and now this is occupying this 3D chemical space. So this is what Schreiber has realized, and he said, okay, making the molecule, large number of molecules is fine, but that has to be stereochemically, skeletally, and appendagely, means they should be diverse enough to occupy all possible 3D chemical space. Then the chances of getting hit molecule are more. Then, like 10 years back, the Chris Abel, he is a professor, still alive in the University of Cambridge. He given another concept. He said, okay, that is also not necessary, making large number of molecules. Let's make only few, but make the best one. So how will you know, okay, the one which we are making few numbers and they're going to be best one? He said, do one approach. Identify few fragments, okay, and small molecule you can think about, let's say 5 or 6 molecule or 10 molecule is a small fragment, a small whose molecular weight is around 300 atomic mass unit, chota molecule. And then screen all those molecules, and out of those molecules you found, okay, this molecule active, but their activity is not good, it's only micromolar range. Now what you can do, these small molecules which were active at the micromolar level, that those molecules you named as a fragment, okay, and then try to connect these. Like a game, you connect the dots and then you become a picture, no? We all do it in our childhood. ठीक है, एक डॉट से दूसरे चिप करते थे, कहीं मंकी का, कहीं कैट का फिगर आता था ना. Similarly, he said, okay, two small molecule, then connect them, and then you get drug molecule. And there is successful story. Actually, those 15-year time, which I mentioned on the second slide, that there is in one case, which I will show you, that within a 5 year, he got a molecule, a very effective molecule, and that is a drug molecule. I currently use a drug molecule through the fragment-based drug design, which is called FBDD, fragment-based drug design. Okay. Very recently, there are another concept also got added up, biology-oriented synthesis. Because everything was going to chemist, then biology, because they are in majority, they think why use only chemistry, there has to be biology. Then there is another concept has come, biology-oriented synthesis. Okay, so these are the few approaches by which people are developing molecule. This concept is clear? Because this is a workshop, this is not like a symposium or something, so I should present my work, I should also, impart some knowledge on you or enhance your knowledge. Is it clear? So now next is, what's the biological mechanism, how the drug actually acts. There are many targets in the biological system, you might be aware about this one. There are receptor antagonists, and this is like, this is a bit old data, almost 10 years back. There are top 50 drugs worldwide, their data has been, compiled, and it was very clear that most of the drugs are enzyme inhibitors. You see this light blue part. So whatever the molecule acting as a drug, although this is almost same by 2025 also, but this particular slide is taken from the AstraZeneca, and data is from 2015. So 38% of the molecule which is used as a drug, they are enzyme inhibitor. Because when we discover any drug act with some mode of action. तो ड्रग अपना काम किसी एक एक्शन के एक मोड से करेगा, रास्ते से करेगा. So the major one is the enzyme inhibition. Next major is receptor antagonist, the 24%. Then we have receptor agonist around 12%. So these three, antagonist, agonist, or inhibitor, these three, they are actually covering major type of mode of action, in the biological mechanism. There are few like some ion channel modulator like sodium potassium pump we are having, those drugs like hypertensive drugs and all. So 8%. Okay, so that means these are the area where of chances of getting successful molecule are more. Next is, what is, by chance discovery, natural product scaffold like Taxol, the clinical observation one. What is the diversity oriented synthesis, which I was talking about 3D chemical space and all, which I am explaining in this particular slide. Let's say what diversity oriented synthesis mean. These are not molecule, these are just like a cartoon, just like a representation. Okay, don't think about it cyclohexane or cyclopentane. Okay, so just think about, let's say this particular molecule, which you can think of having like 6 membered, 5 membered together, and now what you are trying with the same molecule, you are connecting this particular dot, green one, with the red one, then you end up with this molecule. Clear? Like 6 and 5 membered. Same starting material, now you are connecting this dot to this dot, you are getting this molecule, 6, 5 and 5. They are skeletally diverse, ना, skeletally different. इनका जो skeleton है अलग अलग हो गया ना. One is 6 and 5 membered, other is 6, 5 and 5. The skeleton is different, isn't it? The third one, if you connect this dot to this dot, green to blue, then you get this one. This is 4, 6 and 5. From the same precursor, instead of in the high throughput screening or in the combinatorial approach, what people do, one time they put the green dot here, another time they put the red dot here, another time they put the blue dot here, means the skeleton remains same, but they do modification here. But in diversity oriented synthesis, you see what they are doing. They are making different skeleton from the same substrate. So now the chances, because this 6 membered ring and 5 membered ring, when you think about in the molecular framework form, मॉडल में उसको बना के देखेंगे तो उसका 3D केमिकल स्पेस अलग होगा, डेफिनेटली अलग होगा. So this is what Stewart Schreiber has actually visualized or has been planned or thought about it, and he discovered this approach, and by which you make the diverse collection of compound and diverse. This is if you distribute these molecules in this x and y coordinates, and you see they are present everywhere. See, यहाँ पर भी है, यहाँ पर भी है, and this one if you see from the high throughput screening, maybe your molecule are existing here or here. This is not those data, don't get confused with those anti-cancer activity data, na, where the cell. This is simply just a schematic representation that on this 3D space, molecule are everywhere. Then from here, you do the screening and you get the hit candidate and then do the lead optimization. This is another like very systematic representation of like a comparative study of combinatorial approach and a diversity oriented synthesis approach, the below diagram. If you understand this, then I think my job is okay, that what is the diversity oriented synthesis. If you see the below space, what's happening here, starting from the similar precursor, you make the molecule which hitting the similar target structure, and in this 3D chemical space or in this coordinate x and y z, most of the molecule sitting here, only one place, means similar type of structure, not diverse enough. But in diversity oriented synthesis, start from the similar substrate, but you are skeletally diverse so much, and you see in the 3D chemical space, they are everywhere. Is this cartoon or this picture is clear? Any doubt? Very much clear. So this is what I mean by the diversity oriented synthesis, and the detail of this research has been published like in the Nature Communication or Engevante Chemie. They are really high reputed journal. And when the first concept actually came in 2001. This almost also 25-year-old concept. diversity-oriented synthesis. And so that there was a paper in Science. These both paper belongs to cyber, and very much concept was given in 2000, was validation, everything was coming to 2001, long back. And those of you who are interested in this kind of question, write down these references, and you can download these one and you can read about it. Okay. Next is okay. So why there is a case study example. What is the case study? Case study means like what are the molecule which has been made from by diversity-oriented synthesis approach and how they works. So this is the case study example was given by Schreiber's student. He is a professor in Cambridge, and his name is forgetting the Andrew, actually. Yes, Andrew. So what he did, he took the some precursors and made the molecule, and two of the molecules, they were gone to the phase 1 clinical trials, and they are continuing to phase 2 and all. So why we want to make the molecule, as I mentioned, in order to tackle the multi-drug resistant bacterial pathogens, and then target resistant mechanism has to be there, and then search for new antibacterial agent with novel mode of action. These two very important, because if you are making the molecule which act as the same mode of action, so they may not be effective onto the bacteria which develop the resistance. So there has to be different mode of action. And so in that particular work by him, what he emphasized that the small molecule collections, they can come from natural products, they can be from commercial collection also, because by now, how many molecules has been discovered? आज अगर हम 2026 की बात करें, any idea like how many molecules are known so far? No idea. Okay. Exact number I also don't know. Okay, but this is around like a 40 million molecules. This could be very, because exact number I am also knowing. That number of molecules already exist in the literature. They have been made either natural products, or there are so many people around the globe, they are doing PhD, and in PhD, you know, you have to either you to do the new molecule, either you have to do the new biological activity. So people are keep making the molecules, so number of molecules are there. So therefore you can, people are thinking, okay, why should we make the molecule? We can take from those data bank, like commercial collections. And many companies, they are selling those molecules, okay. Not all, but many molecules, they are commercially available. You can go and order from the different company, you don't need to make. So this is another source. And the third one is the diversity-oriented synthesis, what he's emphasized, because he was working those molecules, therefore, in his approach, that molecule has been obtained from the diversity-oriented synthesis. Then, in natural product one, then there is challenges also. And because when you think about taking the molecule from natural products, the very first step, isolating the molecule from the natural product, is a very tedious job. So if you have seen in your lab or in your institutions or here, that if you want to isolate the natural product molecule from natural source, it's not an easy task, because in nature, in one plant, thousands of molecules are there. So separating them, isolating them is a very, very tedious process. So often they come as a mixture of compounds, and also because there are many, so their identification is also a challenge. Like, and then chemical derivatization is also difficult, because when you want to go for the lead optimization, you got the molecule, and now you want to do the lead optimization. So if there is no possibility of chemical modification, then the lead optimization is very, very difficult. So these are a few challenges from the natural product. In the commercial collection, basically there are molecules which have few stereocenters, low molecular weight compounds, and also therefore there are challenges existing in the commercial source as well. Therefore diversity oriented synthesis is one of the best way of making different molecules. This is again this is paper in the Nature 2011 and Nature Communication 2010, where it has been comparatively emphasized that in the diversity oriented synthesis you get skeletal diverts, stereochemical diverts, and appendages diverse molecule. So what is the skeletal diversity? As I had shown, like 5, 5 and 6 type of molecule, or 6 take 5, or 5, 6 and 4 type of molecules. And stereochemical, I given the example of glucose and galactose. And the appendages diversity is like, this is basically maintaining the hydrophilicity and hydrophobicity of the molecule. Let's say you have carboxylic acid group at some position, the molecule would be naturally hydrophilic in nature because it has a carboxylate. Now you think, okay, this more hydrophilic molecule, you want to tune the its hydrophilicity, you want to make it hydrophobic. Then what you will do? Simply do the esterification. Convert the carboxylic acid into the ethyl ester. So now that carboxylic acid hydrophilicity got changed into hydrophobicity. Or install the, do some like amidation, CONH2 group. So this type of diversity is called appendages diversity. So this is possible in diversity oriented synthesis. So here you get, you make all possible compounds and most diverse, therefore getting hit molecule are more. And this is the data taken from the MDL, and this is like you do blue dots. Blue dots is correspond to the drug in general, means all type of drugs, and where they are in terms of their log P value and in the molecular weight, because these are the two very important physical properties of the molecules. Log P, what is log P? Partition coefficient. That basically tells us whether this molecule is going to be absorbed in our intestine or not. Like what is the hydrophobicity and hydrophilicity of the molecule. So lower the log P, polar is the molecule, hydrophilic. Higher the log P, it's hydrophobic. So let's say if molecule is highly hydrophobic, then it won't get solubilized into water. If it is highly hydrophilic, then it cannot cross the cell membrane. So your molecule has to be amphiphilic, okay, optimum. Like amphibians, frog, it can live in water, it can live into the surface. Okay, so so log P is very important phenomena, and this is experimentally can be calculated also. And the software, by software, you can do the theoretical calculation as well, and through experimental also possible, and by using octanol water. Anyway, so what I am trying to convince you, what message I want to deliver with you here, that most of the molecule which has been taken from MDL, what is MDL? What is MDL? MDL is nothing but Molecular Design Limited. There is actually company, and who has the all depository of the molecule. So from those depositories, when the molecule has been screened in general, you see the blue dots, they are spreading everywhere, all the places, very, very much diverse. But when you talk about the antibacterial compound from the MDDR, which is the Molecular Design Limited Drug Data Reports, so most of the molecule you see here. There are diverse molecules, but mostly their molecular weight is around 400 and log P around 2. Okay. But if you see drug in general, okay, there are few molecules here. As you see, the molecular weight is around 600 and their log P is around 14. Okay, there are some exceptions also. So that means if you want to make the most active antibacterial or the bacterial molecule which must have the different mode of action, so we should think about filling this space, means more diverse and various log P and various molecular weight. And that only you can make through the chemical modification by doing the diversity oriented synthesis. Clear? So next is, okay, so this is again in diversity oriented synthesis, we have two different approach. One is like substrate based approach, another is reagent based approach for making large molecule. This is like what is substrate based approach? You have taken the different starting material, use the common reaction condition, and you end up with a different molecule. So in substrate based approach, your substrate are different. Okay, how to understand this? Let's say if you want to make more diverse molecule, but you want to use the same reagent. Okay, if I explain in a very simple way in like household, okay, you want to same masala, but you want to different type of curries. So what has to be different? Your starting material has to be different, isn't it? So this is what substrate based approach is. But the reagent based approach, you taken the same substrate, like, and reagent is different, and you got a different product. That is reagent based approach. In household example, you have one potato, different type of masalas, all different type of recipe you can prepare. Clear? So in diversity oriented synthesis, there are two approach. One is substrate based approach, other is reagent based approach. And these are some like schematic representation just to show you that they are three-dimensionally different molecule. So this is the case history example I was talking about. Actually starting from this particular starting material, very simple, common one, and he made so many different molecules. If you see these molecules in terms of structure, they are very much different, isn't it? So if you see this molecule, this is like a bicyclic molecule, and if you see this is bridged molecule, and these are like heterocyclic molecule, and they all different molecule has been prepared from the same starting material. From where idea comes. Think about nature. Nature makes all different type of molecules, terpenoids, alkaloids, steroids, and their precursor is, have thought about what is the biosynthesis of those molecules? Their precursor is same. From the same precursor, by using different enzyme, different reagent, you have got different molecule. Same is here. So from similar precursors, you could able to make all different type of molecules in diversity oriented synthesis, and then the molecule which has been made from the diversity oriented synthesis by the Waits and Thomas, you see they are occupying more 3D chemical space, the red one and the blue one, and the one which was taken from the focused library, like the one which is commutative approach, they are very much occupying very little space. These are the experimental data. Okay. These are the real data. This is not like hypothetical or something. These are the real data which has been taken from the molecule, which has been done by diversity oriented synthesis, by DOS approach. They are very much three-dimensionally different than the one which has been made from the focused library. And this paper has been published in Chemie. Okay, so this was all about diversity oriented synthesis. I have given much information. Actually, we have the limited time. So next is fragment based drug design approach. So this is another concept in these lines by Chris Abel. Clear? So what is the fragment based drug design? So in this particular design, there are few, let's say here, where you take the collection of molecules, a small molecule. This is a schematic representation of your binding pocket, and this was the molecule. Big molecule has been prepared by commutative approach or high throughput screening, and you see this molecule is binding to the binding pocket, but binding is not tight enough. It's weakly bound. So weakly bound means their activity would be up to the micromolar level or something, that level, or millimolar level. So if the molecule is fitting very much into the binding pocket, that means molecular interactions, non-covalent interactions are very strong, and so their activity would be at the nanomolar range. The stronger the binding, better the activity, isn't it? So what he actually proposed, taking the small fragment, first they are fitting into the binding pocket, and then now what you do, you simply connect these this to this, and this to this. And if you are able to make such molecule, so you started from fragment, and then you connected the fragment, got the full molecule, and their binding affinity would be much higher. That was the fragment based drug design. And through this approach, the molecule is on your screen. This particular gel BORF molecule, this was a beta-RAF inhibitor. This molecule is currently used for the treatment of melanoma, and this molecule has been designed and prepared from this particular fragment, which was azaindole molecule, and this is within a 5 year or 6 year, this molecule came into the market. So this is the case study example. Means these concepts are not just like theoretical one. People have shown that they works practically. So this was the molecule. And there are a few techniques involved in this fragment drug design. One is fluoro-based, like fluorescent-based thermal shift, TS. There is a, of course, when you make the molecule, you need NMR, nuclear magnetic resonance spectroscopy, crystallography, where you do the protein ligand crystallization, then mass spectrometry to find out what the molecular weight of molecules, then surface plasma resonance, SPR technique you need, a bit expensive. Then you need in silico screening, like computational modeling and all. Then this one, very important, isothermal titration calorimetry, which is ITC. This is one like physical chemistry analytical actually method by which you see how strong your molecule is binding. You see whether it is binding weakly or strongly. When you connect, then okay. So ITC is one very important technique which is helpful to see whether your molecule is interacting with the protein or not. So these are the some techniques you need for the fragment-based drug design, and this concept was given around 2010 or 9. This also again 10 to 15 year old concept. The Nature Chemistry paper has come in 2009, and Nature Chemical Biology again by the same group. So where they have explained this particular concept and also development of this molecule through the fragment-based drug discovery. So now we have compared the HTS versus DOS. Now this is time to compare between the fragment-based drug design and diversity-oriented synthesis. What's the difference is? In fragment-based, I think this is a very beautiful slide. I hope you all understand. This I already shown you. Okay. In diversity-oriented synthesis, let's say you have intermediate or substrate. Now you are connecting, what you are connecting? You are connecting this dot to this, you are getting this molecule. And you are connecting this one to this, you are getting this molecule. And if you are connecting this to this blue to green, you are getting this one. Is it clear? From the same substrate, you got different, three-dimensionally different molecules. This is diversity-oriented synthesis. But what is the fragment-based drug design? Fragment-based drug design is not believe in the connecting all those dots, and rather what Chris Abel proposed, that take a small fragment. You think about, let's say this one molecule, this is another molecule, this is one molecule. You screen all these molecules, small molecules, not the one which has below 300 molecular weight. And you got out of these three, you got low affinity lead molecule. Low affinity means it's active, but its activity is not that great. Low affinity. But this as a lead molecule. Now what you have done, you started optimizing one, little bit decorating it. What is the philosophy of starting with a low molecular weight? Because you have to, later stage you have to do modification. Okay, and so you started from 300 atomic mass unit, got lead, and now when you are doing the modification, your final molecular the molecular weight of final molecule reaches up to 500, and that was the optimum molecular weight given by Lipinski in 1989. So then from here you did the high affinity compound, then you do further optimization, and you got the drug candidates. So this is how both approaches varies, and both are very effective, and they have the successful case study example. So now you might be thinking, okay sir, which one is better? तो obvious question आता है ना, ये भी है, तो उसमें से कौन अच्छा है? So if you ask me this question, then my answer is very simple, that this particular approach is more effective because here you don't need to make large molecule, rather few molecule, and the chances of getting hit. Diversity-oriented synthesis, when you are not getting hit, then we have to make diversity-oriented synthesis. But obviously both are superior than those traditional way of making molecule, large number of molecule through combinatorial approach. ठीक, उससे तो दोनों बेहतर हैं. Clear? I hope I have given you the ideas what is the fragment-based drug design. Clear? यहाँ तक. Now let's come to the, and on these lines, actually my laboratory here at JNU, and my school is next to this building, School of Physical Sciences, and Yasika actually, she is the intern from among you, is working in my lab. So we are making the glyco-hybrid molecules. So in glyco-hybrids, what we are doing, we are identifying the privileged scaffolds, and we are connecting to the carbohydrate molecules. So, okay, these are few another slides where in hit library you make this big ball, a big balloon means these number of molecules you have to make, then you get this number of hit. So lot more resources needed. But in combinatorial approach, you made little molecule, and you do optimization, you get drug candidate. So this less molecule is needed, more molecule is needed, and the ultimate goal is same. These are the actual case study example how in fragment based drug design, by connecting these two fragments which were active at 100 micromolar, which is not very good activity, drug molecule has been modified. For example, this was 100 micromolar, then by taking this as a fragment which is exist here in this part of the molecule, and then they did little optimization, and then they make this final molecule which was active at from 100 micromolar up to 6.5 micromolar. Almost how many times? 20 times better activity. 20 times, isn't it? And then if you do further optimization, you end up with the nanomolar as well. So, and this all details, there is a paper in Journal of Medicinal Chemistry published. You can note it down and you can see the story of this one. And this is another one like drug discovery today. This is one slide I want to show you to just give the comparative analysis or comparative study of, because in pharmacy or in the biology or in chemistry or in medicinal chemistry, we always teach like Lipinski rule of 5. What is the Lipinski rule of 5? Anyone? Lipinski rule of 5 सुना नाम? नहीं पढ़ाया गया? So Lipinski rule of 5 says, which was actually given by Professor Lipinski in 1989. Yes, 1989, if I am correct, not 98. 1989. So Lipinski said, the druggable molecule, the one which can be potentially a drug, molecule should not be above 500 atomic mass unit. इसका molecular जो weight है compound का, वो 500 के आसपास होना चाहिए. Lipinski ने कहा. Just like you can think about in society, if you want to be a good runner, आपको कोई दौड़ जीतनी है, तो आपका weight एक fix होना चाहिए. अगर आपको 1000 atomic mass unit का molecule है, तो druggable नहीं होगा. There must be the log p, partition coefficient, उसका 5 के आसपास होना चाहिए. This has to be around 5. There was second law, and third was
hydrogen bond acceptor and donor. Hydrogen bond acceptor and hydrogen bond donor.
Hydrogen bond acceptor and hydrogen bond acceptor has to be 5, and donor has to acceptor 10, and donor has to be 5. Okay, so this is also multiple of 5. And 4 हो गया. पांचवा क्या था?
Molecular lipophilicity.
That already been done. The number of rotatable bond should not be more than 5. When in the molecule there is some bond or rotatable bond, some bond or fixed bond, rigid bond. So if there are large number of rotatable bond, that means molecule is very flexible. If that molecule is very flexible, it cannot be a drug. If molecule is a very rigid, उसमें कुछ भी rotate वाला नहीं, बस बिल्कुल जाके एक ही जैसा है, that cannot have fit. Same in the society. If you see the administrator, if administrator is very tough, he cannot survive in society. If it is too flexible, then also people make fun of him or her. This has to be balanced, isn't it? So, same with the molecule. If molecule has many rotatable bond, it cannot become a drug. If it is too rigid, it also cannot be drug. It has to be 5 rotatable bonds. So these are Lipinski rule of 5. But what's the fragment directed design said? The molecular weight has to be around 300 fragment ka, and there are only few hydrogen bond acceptor and donor. Lipophilicity is around, should be around 3, and low to high affinity of the target receptor. And these type of molecules, they act as a lead-like behavior. Where the Lipinski rule says, if they follow these 1, 2, 3, 4, where is the 5? Rotatable bond is not here. Okay, so there is a 1 rotatable bond also there. So there is a drug-like behavior. Clear? Now I will go to my work actually. This is in next 45 minutes I will present glyco-hybrids. So what are the glyco-hybrids? Those of you not aware, so glyco-hybrids are the molecule which are formed by the combination of privileged heterocycles. Privileged heterocycle, what is privileged? The one which has the potential biological function or natural products, like privileged person, very important molecule, connecting with the carbohydrate derivatives, and all together these one gives the molecule like here or new biologically active glyco-hybrids. This is the hybrid molecule of carbohydrates and privileged heterocycles. And while doing so, we developed several synthetic methodology, because when you make the molecule, there has to be method. Because if you are making the new molecule, totally new molecule, so then you think about some new synthetic approach, isn't it? So we make the molecule, we look their biological function, and we publish in a biology related journal, and we do the methodology by combining this, and then we publish in the chemistry related journal. So like one work, 2 results, 2 different type. Clear? So if you understand this triangle, and okay, so why we want glyco-hybrids? Okay, I'm making glyco-hybrids, fine. I have designed the glyco-hybrids, fine. But why then? Why? Why it is necessary? How does it helpful? So I think you all agree with me that when you have carbohydrates, the carbohydrates molecule are natural molecule. They are not toxic because they are edible molecule. We eat the glucose and galactose, fructose. So when we attach the carbohydrate to the molecule, very first thing what it does, it reduces the toxicity. It decreases the toxicity of the molecule. This is value addition. It increases the solubility because this has many hydroxy group. The solubility of the molecule is improved, has increased. Clear? Solubility got increases, toxicity got decreases, and this is what we need for the medicinally active molecule. It has several chiral center, and in our body or in our protein, they are all 3D molecules, 3-dimensional molecules, chiral molecules. All 20 amino acids, out of 20, 19 molecules are 19 amino acids are chiral amino acids. Okay, so they make the 3 chiral environment, and these molecules are going to interact with the protein and enzyme. So chiral interaction, like likes dissolve likes. So chiral molecule, the chances of getting interaction more. This is another advantage. The one which is very much advantageous is that when you have carbohydrate attached to your molecule, its selectivity may improve. I will give one example which is very much I think taught. I don't know whether you have been taught or not. Something called Barber effect. WARBURG. What is Barber effect? Anyone? हाँ, क्यों करने लग जाते हैं पर? एनर्जी किसने बोला? हाँ, the cell need more energy, more ATP. You are also correct. So why they need more energy? Because they have to divide, mitotic division. ठीक, therefore when there is any tumor or part of the body, you touch it, you see that's temperature almost to 1 or 2 degree higher than the normal one. Even जो simple फोड़ा होता है, फुंसी होता है, वहाँ आप छू के देखिए गर्म होता है, because their metabolic rate is very high, because they need more energy to divide, to multiply. So a source of energy for human, for us like vertebrates, is the glucose, glycolysis and ATP. So they need more energy, therefore they need more glucose molecule, therefore the GLUT receptor are overexpressed, so they takes more glucose intake. So when you connect your molecule, active molecule, with a glucose moiety, the chances of that molecule reaching to the cancer site is much higher. This is what Barber effect is all about. So this is what I was talking about, selectivity. When you connect these sugars moieties, so there are chances that they may selectively go to the disease site, target site. Therefore, because of these 4 or 5 advantages, making glyco-hybrids is a wonderful research. This is not wonderful because I am doing, but it has a reason. So these are some examples, and you see this particular molecule, this is like for biology people can also recognize this. This is 4-hydroxyquinolone. This is a privileged scaffold of this category. And now this is the carbohydrate moiety. It's not very similar to glucose, but it has all skeleton, everything same, but double bond is here. And this is we call 2C-pharmyl glycal. And we connect these together and we got new molecule, which is pyrenoquinolones, like tetracyclic 1, 2, 3, 4 cyclic molecule. And this is the novel molecule which has this part, 4-hydroxyquinolone part, and which also has the sugar part. Clear? And this molecule we have done, actually made a few years back when I was in faculty at Banaras Hindu University, BHU, and this particular molecule was found active against breast cancer cell line with IC50 value of 3.53 to 9.68 micromolar range. Not very great activity, but this is better than what 4-hydroxyquinolone alone is having the activity. So that means when we added the sugar moiety, their activity has been improved. This is one case study example. Another one, if you see here, this is also now almost 6-7 years back story. We made some furocoumarins analogs starting from 4-hydroxycoumarins, and these 3 molecules were found active against the cancer cell, and their activity all done in collaboration, because I am the synthetic organic chemist by training, medicinal chemist. I can design the molecule, I can think, okay, this molecule could be better, this cannot be better, how to make these molecules, but the activity has to be done with the biologist. So whatever activity I am presenting here, this was all done in a collaboration with different. Then we have this is also a very good example of glyco-hybrids, because if you see this part, blue part, is it visible? I think there is one student sitting behind watching the mobile. Can you please pay attention here? Is it not interesting? So this is 4-hydroxycoumarin, and we connect this 4-hydroxycoumarin with this sugar moiety, the pink color, through a linker. This is triazole linker. It's very easy to make by click chemistry. Alkyne azide chemistry, and that chemistry has been awarded the Nobel Prize. Professor Caroline Bertozzi, K.B. Sharpless, and Meldon. These 3 people have got Nobel Prize for discovery of making this triazole ring. Very simple thing. Anyway, so this is the link glyco-hybrids you can see, because there is a linker, triazole, sugar moiety, and this was again published in 2019 and having anti-cancer activity. Then this is very interesting one. You see, this particular skeleton, this is called carbazolone, and if you blue part is a blue part of this molecule, जो इसमें blue part है वो क्या है? What is that? बायोलॉजी वालों से क्वेश्चन है ये. जो ब्लू पार्ट का हेटेरोसाइकिल है उसका नाम क्या होता है? बेंजीन रिंग, उसके बगल में 5 मेंबर एक नाइट्रोजन का रिंग. हाँ, वेरी हाँ नहीं, उसने बताया कुछ. बोलो बोलो, तुमने कुछ सही बोला, तुम चुप शर्मा क्यों रहे हो? हाँ, पिरिमिडियंस होते हैं, उससे एक ही 6 मेंबर्ड में होते हैं, 2 नाइट्रोजन होती हैं. इसमें एक हमारा बेंजीन है, बगल में 5 मेंबर्ड में नाइट्रोजन है. ये बता देगी. बहुत सिंपल सा मॉलिक्यूल है. अभी हमने उसका नाम भी लिया था उधर जब फ्रेगमेंट बेस्ड डिजाइन पढ़ा रहे थे, बता रहे थे. चलो केमिस्ट्री वाले बताएं. इंडोल, इंडोल रिंग. ओके, है ना? Correct? So this indole moiety, when connected with the sugar part, this is called carbazolones, and these carbazolones and carbazoles, they were prepared from the starting material. So if you see, this is privileged scaffold, blue part matching with this blue matching, and this is the sugar part, this red one. Okay, so all together they are called glyco-hybrids, published in organic letters, which is very high impact journal in the area of chemistry. Anyway, so if you understand this slide, then my job is over. Okay, the today's because I made 3 objectives for today's presentation: to explain you what is the drug discovery process. Remember, means to just revise the drug discovery process, to give the ideas about the diversity oriented synthesis, to give idea about the fragment based drug design, and give idea about the glyco-hybrids. Okay, so now what I am going to present, the 3 stories, very small stories, the real work of very recently the one glyco-hybrid which we have, my laboratory, my students has made in JNU. Are you ready for that? Okay, so these are the glyco-hybrid structure which is currently used as a drug. For example, you all familiar with this molecule, I hope many of you, doxorubicin, anti-cancer drug, है ना? So if you see this molecule, this red highlighting about privileged scaffold, and this part here, this is a sugar, amino sugar. Okay, not amino, rather amino. Okay, so this is amino sugar. If you see this part, this is anti-diabetic molecule, currently used a drug, where you have this benzothiazole, and then we have sugar part in it. This is glyco-hybrids. This is bengazoles, where you have these heterocycles. These heterocycles are oxygen, nitrogen, oxazoles. Oxazoles connected with the acyclic sugar moiety, because sugar can be cyclic form and sugar can be acyclic, Fischer projection form. Okay. So this is also glyco-hybrids, and this is the antiviral molecule, like this is marivavir molecule, which has the privileged cycles and this particular glyco-hybrids. And these are the few glyco-hybrids which my laboratory actually made in the recent few years in JNU. Actually, this is from BHU, and these one are recently, because I am here since 2020, almost 6 years now. So, so these one, like OBC paper in 2025. This is the sugar moiety. This is the privileged scaffold. This is organic letter 2025 made by Yogesh Yadav, and here we have indole and this acyclic chain. Very recently, December 2025, this furocomerins made by Preeti, and then this one by Ghansyam, where we have pyrimidinones with the sugar moiety, and this one by Ghansyam again, and this is by this particular like molecule, like imidazopyrimidinones. So this was recently made by Neetu. And so these are few glyco-hybrids which we have made recently. Actually, few are not included here. From, is there even from 2026? No. So we have made in 2026 as well. And these, how they have been made. This is very actually this particular slides, only one slide, but there are 4 or 5 stories. If I start explaining one by one, it takes another like 2-3 hours. So I am just giving the glimpse that we have made few glyco-hybrids starting from very simple precursors. This is pyrimidinone. Pyrimidine structure with a ketone, with a sugar, and we got this cut of molecule published in Journal of Organic Chemistry 2024. And then this is two alpha-ido-enone derivatives were coupled, got this type of molecule. And very importantly, because one very important thing in medicinal chemistry, we must remember that our method has to be simple. Because if you are making molecule and if your synthesis is very complicated, very expensive, so that's going to not work in longer run, because your method has to be very simple and it has to be metal free. Because if you are using very toxic metal in your synthesis, for chemistry is okay, because you do the column chromatography, you took the NMR, you said there is no impurity, your compound is pure. But when it goes to biology, even traces amount of metal, it gives a very false negative result. False positive, not false negative. Okay, and also it causes toxicity. Maybe your cells are getting killed by that particular metal which is present in a nanomolar quantity in your sample, not by your molecule, and you are claiming that you have made the anti-cancer molecule. So method has to be simple and it has to be metal free. And if you see these processes, no reagent is in fact needed, only ethanol is a solvent. Either you thermally heat 80 degrees centigrade or you heat in microwave. In microwave 5 minutes, at thermal heating it takes 4 hours and you get 80 to 60% yield. And these are some advantage you can, chemistry people can understand better, that we have achieved all those things. And then if you see this one, only potassium carbonate you need as a base, because potassium carbonate everywhere, not very expensive. Even in MSc undergraduate lab, you go and get the potassium carbonate. So those type of reagent we have utilized and successfully made those molecules and published in very high impact journal. And not only that, the precursors, the starting material which we are using, if they are very expensive, because especially in our country, we have to think about simple, there's a quotation called, I'm forgetting the quotation, but you think as simple, but you aim bigger. Okay. Similarly, this one, 2-hydroxynephthoquinone, if you see pyrrolidine as a reagent. Pyrrolidine is like a secondary amine, it's not very expensive and also not metal. And by using simple pyrrolidine, you are able to connect these one. And you got this very beautiful molecule which is structurally very similar to doxorubicin, which has the privileged scaffolds, which has the sugar moiety. This one doing the copper chemistry, copper relatively safe as compared to other metals. This was work done by Neetu in Bioorganic Chemistry 2025, where she has beautifully made these pyrimidine scaffolds connected with the sugar, with the triazole ring, along with the one appendageous diversity. It has the stereochemical diversity, it has the skeleton diversity, it has the appendageous diversity. Okay, and also because of this chiral center is stereochemical diversity. So all those things which I was teaching about in diversity orientation, all those parameters are fitting in this one molecule. And then one point I also emphasized during my presentation, that once you got the molecule as a hit molecule, then for the lead optimization, your molecule should have some chemical functionalities which can further get modified. Then only you can do lead modification. आपने ऐसा molecule बना दिया जो उसमें कोई आगे chemical reaction possible ही नहीं है, तो आप lead optimization कहाँ करेंगे? Just like you have made house and you haven't left any space, or you haven't any you want to extend your house, but you haven't left any space. Can you extend it? No. Same with the molecule. The molecule, the one finally you have designed or made, it must have some scope for modification. But these one, the one which I am showing here, they have a lot of scope, because you see so many hydroxy groups. You can play with these hydroxy groups. And if this R is something, let's say OH and all, here you can play here. You cannot play much with this skeleton, but there is a lot of scope playing here, here, and especially this particular triazole ring. So These are another glycohybrids which we have made. We have made a method for making 2-deoxy sugar. Then by using that one, we made these bis-indole analogs last year. And very recently we have made these indole C glycosides, and we have done very fundamental observation during this. Lot of chemistry is there, and where in one method we got one type of glycoside. If you see structurally, if you see this one, this is glycoside because connected to the anomeric position. But if you see this structure versus this structure, what is the difference? The difference in the yellow part. ये तो इसमें भी है, the red one here also, and this is here also, but yellow part is not here, isn't it? So what actually, there is a chemistry, lot of chemistry is here. So when we have electron withdrawing group on the indole ring, we were getting this molecule. When there is electron donating one, we were getting this one, and we very systematically analyze why we are getting this one. Because in science, science is not like simply because we can do it, that therefore we are doing. Science ऐसा नहीं है कि हम कर सकते हैं इसलिए कर रहे हैं. Science has to be why we are doing it. There has to be very strong reason of doing that. Why? What's called 4W 1H? There is one very specific quotation. How means how you are going to do the science, why you are doing it, what you are doing it, when you are doing it, and where you are doing it. This is called 4W 1H. This is very important in life. Similarly, in the academician's life as well, like us. Why we are doing it, that is very important. One is okay because we can do it, therefore I am doing. No, that's not enough. There has to be a specific reason. So those reasons I tried to find out in my chemistry. Actually, I did get few answers. So these three stories which I was talking about, one is about making this quinoline glycohybrids, was done by Kavita. Then this one done by Rajdeep, and another one very recently, so this one by Preeti. So this is December 2025 story. They are not very old, December 2025. And this one February 2025, this one is March 2025, a 2-month old. So what we have done, this is a paper published in JOC. Those of you who are interested, you can note it down this reference and go in detail how these molecules are made, and done by this particular PhD student in my group. And why we were interested in this class of molecule? Because nitrogen containing heterocycles, they are considered the privileged scaffold. Now, my question to all of you, why the nitrogen containing molecules are druggable molecule? Nitrogen वाले ही molecule drug ज़्यादातर क्यों होते हैं? Any thought, any idea? या जो आपने सीखा हो अभी तक?
Because they can bind to the glycoside through the N-glycosidic bonds.
N-glycosidic bond. What he's saying that because they have the nitrogen, so there is actually not glycosidic bond, there is a hydrogen bond, okay? Because the interaction between the molecule to the protein happens through the non-covalent interactions. Van der Waals interactions, hydrophobic interaction, pi interactions, and hydrogen bonding interaction. Hydrogen bonding is the common. Hydrogen bonding, okay, this is one. Any other idea? कुछ ऐसा बताइए जो लगे कि हाँ मतलब ये हो सकता है. हैं? ये कुछ बताना चाहती, बताओ. Why most of the drug are nitrogen containing heterocycles? Any idea? To stability of the molecule, but there are many stable molecules, so all stable molecule cannot be a drug. Nitrogen has very specific reason actually, and this actually I couldn't find any like the most standard book for the medicinal chemistry is Graham Patrick, na? How many of you read that book, Graham Patrick? नाम सुना है कि नहीं? This is available nowadays. You are Gen Z, you know how to download it from, it's available online also. If you know, I can give it to PDF file. Graham Patrick was very standard medicinal chemistry book. Okay. Those of you from are interested, make your career into the biology or medicinal chemistry or drug discovery, that is the reference book. पढ़िए आप लोग, ठीक है? So why most of the molecule are the nitrogen containing molecule? Because as you've given the full form of the ADME T in the very beginning, so first step is the absorption. So what that phenomena is all about? Absorption कहाँ होता है body में? Which part of the body the absorption took place? Small intestine, okay, through the microvilli. So small intestine, the absorption happens. Then molecule from the from those intestine reaches to the blood. So transfer of molecule from intestine to blood is called absorption. Next is distribution. What is distribution? Through the blood. ये कुछ बोल रही है क्या? You said something. Yes. So distribution is not like that from blood going to the brain, hand, leg. That is circulation. Don't get confused between distribution and circulation. Generally people think okay, distribution यहाँ से इधर चला गया, distribute हो गया. Circulation हो गया वो. Distribution means transfer of molecule from blood to the cell. As he said, target. सेल के अंदर ब्लड से सेल में जाने का जो प्रोसेस है, टिश्यूज में जाने का जो प्रोसेस है, दैट इज डिस्ट्रीब्यूशन. व्हाट इज द पीएच ऑफ ब्लड? 7.4. Slightly alkaline, 7.4. If your molecule is, let's say, acidic, then it will try to remain into the alkaline medium. ब्लड में रहना चाहेगा ना, बिकॉज ये एसिड है, ये बेस है, साथ साथ रहेंगे साल्ट फॉर्म में, घूमेंगे. कीप सर्कुलेटिंग इनटू द ब्लड, गोइंग अगेन इनटू द लीवर, causing damage to the liver. So when your molecule is slightly basic in nature, nitrogen, what is the nature of nitrogen? Basic. Then the distribution process will get facilitated. Transfer of molecule from blood to the cell is, so nitrogen helps actually. It definitely helps what somebody tell here, hydrogen bonding one, okay, he has tell, but it also helps into the distribution.
So there are many other molecules which is basic in nature, so why we can't use them?
There are many other factors which actually stopped using them. It's not, as I said, it's not necessary that all basic molecule can become drug, but most of the nitrogen containing molecule are drug. So nitrogen basic nature helps into the distribution. Absorption, distribution. Third is, what is metabolism? And why it is important? फुल फॉर्म तो सोशल साइंस वाले भी याद कर लेते हैं. साइंस वालों को वाई. व्हाट इज मेटाबॉलिज्म? मेक द वो तो अब पहले बनाएंगे ना, तो ड्रग तो अंदर चला गया बॉडी में, अब उसको पोलर नॉन पोलर से हाँ दिस इज ओके, बट क्यों, वाई मेटाबॉलिज्म इज रिक्वायर्ड? हाँ व्हाट इज बीएमआर? बॉडी बेसल मेटाबॉलिक रेट. हाँ वो तो ठीक है, वो आपने बता दिया मेटाबॉलिज्म क्या है, लेकिन क्यों, वाई मेटाबॉलिज्म इज एसेंशियली रिक्वायर्ड? पोलैरिटी चेंज होगी, लेकिन पोलैरिटी चेंज होकर क्या फ़ायदा मिलेगा? एक्टिविटी बढ़ जाएगी.
टॉक्सिसिटी से बचने के लिए बार-बार डोज ले रहे हैं तो उससे मेटाबॉलिज्म करके बॉडी से एक्सक्रीट आउट करना है. तो
हाँ ये बात है, अब आप पहुँचे वहाँ पे, देखा. सो दिस इज हाउ यू कैन एज ए टीचर यू कैन गिव द थॉट्स टू द आंसर. यहीं से आ गया ना. Metabolism is required because if the molecule remain in your body for over very long period of time, it going again and again to the liver and it causes toxicity. So then this has to get metabolized and get excreted out through urine or through sweating. Okay, so metabolism is required in order to clear out the molecule from the body in order to minimize the toxicity. Therefore metabolism is required. इसलिए metabolism के बाद क्या है? Excretion है, फिर toxicity है. जो ADME कोई उसका सीक्वेंस है, ऐसा नहीं कि उसको TAMD क्यों नहीं बोल दिया, because it happens one by one. And one thought came from here that that is become polar and become drug molecule, and that is not completely true, but partially is okay. I think she has said, because there are few molecules which are pro-drug, means they are not drug in their that form what you are taking, but once they inactive drug, but once they reach into the blood, they get little metabolized and they started acting as a drug. Any example?
Promtosil. Promtosil.
Yes, another one. There is one pyrazinamide, I think tubercular drug, that is also pro-drug. So metabolism, okay, so most of the nitrogen containing drugs, because they help into the distribution, and therefore these molecules are nitrogen containing, what we have aimed. And in this, while making this molecule, we have started from very simple precursor, chiral synthone, and this chiral synthone is nothing but the glycan. See, many of you are familiar with this particular structure right now, and this particular molecule has been utilized to make these sugars, 2-deoxy sugars. What is the difference? What is T-deoxy sugar? At 2 position, numbering start from here, 1, 2, 3 and 4. The 2 position is, you see, there is no hydroxy. इसका 2 पोजीशन पे जहाँ रेड डॉट लगा हुआ है वहाँ पे हाइड्रोक्सी नहीं है ना, वो मिस्टेक नहीं है, सही करेक्ट स्ट्रक्चर है ये. Therefore this is 2-deoxy sugar. So 2-deoxy sugar are very much useful. 2 molecule, you tell me what are used as a drug as a 2-deoxy sugar. One, I am giving you some clue. One molecule which is 2-deoxy sugar used as a PET imager. PET समझते हैं ना, पॉजिट्रॉन इमिशन टोमोग्राफी. जब ट्यूमर होता है कहीं बॉडी के डीप इनसाइड सेल में, टिश्यूज़ में, उसको इमेज करने के लिए यूज़ करते हैं. एक ऐसा मॉलिक्यूल है जो अभी भी यूज होता है पेट इमेजिंग के लिए. अब मॉर्निंग में जाएंगे, उस सुबह मॉर्निंग में बनाया जाता है, केमिस्ट आके उसको बनाते हैं, फिर उसका हाफ लाइफ रेडियो लेवल्ड होता है, हाफ लाइफ उसका 2 घंटे का होता है. मॉर्निंग में बनाया और तुरंत ही पेशेंट पहले बुला के बैठा लिए जाते हैं, इंजेक्ट दिया और फिर मशीन में लिटा के उसका इमेजिंग किया जाता है. फ्रेश बनाया जाता है बिकॉज इट हैज़ हाफ लाइफ, रेडियो लेवल्ड. 2FDG, हैव हर्ड अबाउट दिस नेम? 2-fluorodeoxyglucose. So 2-fluorodeoxyglucose is one of the 2-deoxy sugar where you have everything is same. At 2 position there is a fluoro, and that all molecules they have isotopes. तो वो उसका F18 isotop है, and that is used as a for imaging purpose. Another molecule which has come in very much in picture during the COVID time, 2DG. News में था ना, DRDO has made a molecule to control the, because during the COVID time maximum death was happening because of suffocation, because oxygen level was oxygen level into the blood was dropping down, and when it reaches to 30, people start feeling problem to breath, and below 30, 20, 25. This was the case. So what's DRDO actually, there is one professor, Anil, and what he found out, he actually repurposed. The 2-deoxyglucose is a well-known molecule since 1980s, but what he found that if the 1 gram of the average 50 kilo body weight of the person, if 1 gram of glucose, not glucose, deoxyglucose is given orally, then the level of the oxygen into the blood somewhat remained balanced. And many people actually have survived with that molecule. And that time there was a shortage. People also contacted me, can you make the deoxyglucose, because I am the carbohydrate chemist, because that time labs was completely closed. So even we cannot come to the lab. Okay. Anyway, so that means those of you, because many people say pure sugar cannot be a drug. Partially it is true. So then why you are working carbohydrates? I give the 2 example, 2-deoxyglucose and 2-fluorodeoxyglucose. They are biologically useful molecules. So the carbohydrates can be useful molecule as well. So therefore, making this class of molecule, very important task. Many people have developed different methods to make these molecules, let's say trifluorotic acid and anidosuccinamide and bromosuccinamide, okay, and there are many one. And we also develop a method for making these 2-deoxy sugars by very simple reagent, trimethylsilyl chloride and in 1,4-dioxane, little higher temperature. Reaction is finishing 1 hour, and if you see here, 36 hour, also this is also 36 hour, 16 hour this one, and this involves 5 steps to get molecule. So all complicated process. And actually this is also our process developed by Yogesh. Within 1 to 2 hours, we were able to make these 2-deoxy sugars. But here what we have used, we use the ceric ammonium nitrate, which is the cerium, which is also metal. But then we thought we were interested to make like metal free, and then we got eventually one method by using trimethylsilyl chloride. We can make the same molecule very simple way, and this one we utilize for making these heterocyclic molecules. So this is one story, and how much time do I have? Is over? How much time? 5 minutes?
11:21.
11:20. So we have 10 minutes, no? No.
Till 11.
Time is over, okay. So time is over. So okay, let's summarize. So I will not go in all details. Those of you interested in this slide, I can share, no problem. So all different sort of molecule we have made. This is another one which is done by this gentleman. He developed a method for the indole C glycoside analogs, and that story published in European Journal of Organic Chemistry this March 2020 around Holi. So my Holi was very much colorful. We got a good paper in the EGOC. And then third one, the third story, we have made this furocoumarine analogs, that was February 2026 by Preeti, and that was very challenging molecule, very challenging molecule, because the molecule looks on the board very simple, but when you try to construct them, you face a lot of challenges. Okay, the yield is not getting good, selectivity is not coming, molecule is, because when you do reaction in RB, this is mixture of chemical is there, so all mixture can go and interact anywhere. Okay, this is not, so we are not doing flow chemistry. Another area also emerging very much in pharma industry, flow chemistry. Flow chemistry, okay. So what is flow chemistry? Like you are doing, you are flowing one reagent one by one, so there is no chance of mixing others. So selectivity is better. So these furocoumarines molecule we have made very recently. And also one important thing I must emphasize, when you are getting any selectivity in chemistry, okay, this is forming, this is not forming. So you cannot simply say, okay, in this condition this is forming, in that condition that is forming. That is not enough. There has to be reason why. Why this particular group, when it's there, only this product is forming. Why other group is forming, why this is forming. There has to be solid scientific support. So in that particular case, we have done the DFT calculation in collaboration with Chhatresh, and then we were able to know why, when there is electron withdrawing group, this is the product distribution, electron donating one, this is the product distribution. Substrate is almost same, reagent is same. In one case, other product, other case, other product. And this happens in science. But we should know the region. That is more important. And last one, this is by Preeti, and she has made furocoumarines. And why furocoumarines? Because they are important class of molecules. You see all different, the blue, the one green and pink. So this furocoumarine part, this one, this the blue part, the furo, 5-membered ring, and this is furopyrene basically. And all together, if you consider this, all here, this is furocoumarine. And these are natural products, like cholesterol is a natural product. This is also natural product. This is HIV protease inhibitor one, and this is this is furcoprolone molecule. This is also natural products, and so therefore in my title I have given the natural product inspired or natural glyco-hybrids based on the natural product scaffolds. And if you see here, this is the furocoumarine, and we have also attached the sugar part to it. So this class of molecule we have made successfully. It was very, very challenging because 3 chiral centers are already there, which I am taking from sugar, but 2 new chiral centers are generating. This is also chiral. This particular center, if you see here, this is chiral and this is chiral. So 2 new chiral center was coming. Is there any? Yeah. So see, these 2 new chiral centers are forming. So if there are 2 new chiral centers are coming, how many possible isomer? 2 to power n. Now, if there is a 2 new chiral centers are coming, if there is 1 chiral center in molecule, how many isomer is possible? 2. If there are 2 new chiral centers coming, how many isomer are possible? 4. Out of 4 possibilities, we were getting very highly selective, highly diastereoselective only 1 product, and that we have confirmed through the NMR. And finally, he was fortunate enough, got the crystal, and so we established the structure by crystallography data. And in like next 30 seconds, I should show you where I am situated in this SPS. So these are the contributors for today's presentation. So this was my lab. Actually, this is my lab, not was. And this picture was taken in 2021 when my lab was established. This is fully funded by, I found, one person, industrialist person, because I moved here in 2020, and that was a time of COVID. And government was putting money into the healthcare, and there was very little money to the academia. And the basic fundamental things, this is for like information, when you go as a faculty member to the new system, university, you get a space, but your lab you have to build up by your own. In India. Yes, in the few institutions where you get the lab, which is furnished one, like IITs or ISRs, but in most of the university system, central universities, or even in state universities, even it is hard to get the space. ठीक? Space के लिए भी wait करना पड़ता है. But I am not the person of kind to wait longer, so I started looking options. So because I moved from the BHU, Banaras Hindu University. So Banaras Hindu University, how many of you know Banaras Hindu University? अच्छा 4-5 लोग जानते हैं. So Banaras Hindu University was established by Mahamana Madan Mohan Malviya ji through the donation. Complete donation. Started project in around 2005 by not 2005 by 1905. Okay, 1905. And by 2016, on Basant Panchami, that university was become functional. And 1905, that time what was the era? किसका शासन था इंडिया में? British. So in British time, if you do something like even this much students are sitting here and I am teaching, so there is a like always okay, ये बच्चों को भड़का तो नहीं रहा है. Means he was not making people to fight for the independence. That was time, and he was going around. So anyway, cutting long story short, so Banaras Hindu University was completely made from the donation. So that was the place where I am coming. I was a faculty, so I know how to take the attract the money. And then I found the Saint Sagneria, and I spoken with him. And the lab also, he was kind enough to support the lab furniture. And here there was no provision actually, because this university has been very rich since long time. Now it's not that good situation, to be very honest. But this place was is very rich. So, but 2020 I contacted this particular person, Saint Sagneria, and he was kind enough to donate the lab furniture. This is how lab has been built up, and then this is these three fume hoods where the Yasika is working, and then this is the first batch of chemical arrived around August 2021. By then, this lab was inaugurated, and I was very much happy. See here, I can do good science, and my son is standing. He's taller than me actually, so I feel sometime bit inferiority complex because he's taller than me, next to me. And then these are this is our honorable Vice Chancellor, and she has come to inaugurate. And this, because there is a purpose of showing this slide. This slide is not like to tell any story. This slide is just to purpose to acknowledge the guy, acknowledge the gentleman who has actually supported for this good cause. And that person is standing here in the dark color, he is a centenarian. So this life is because of him. Because the COVID time, so many people also wearing the mask. So he is the person who donated this all lab furniture. And she is the secretary of this businessman. So she has come along. You recognize this person? See, so we are friend also. And then Dean, and because that was summer, not that summer, but and then our honorable Vice Chancellor Santi Sriji, and then this is the first batch of a student, and almost all gone. She is also going, like she is the Kavita, and and so these are the first batch of a student. She was master from Odisha, master student in chemistry, and then first visitor Mark from Germany during winter time. Yeah, so this is the rotabab, which the very first rotabab has arrived in this laboratory. Not very expensive, because this is not, I am not the ambassador of Aditya Scientific, but this is the rotapaper which I have purchased, and this cost is not very high, around only 3 lakh. And if you buy the same from the Hedolf or Buki, which is like Swedes and German companies, you get in 7 lakh, 7 or 8 lakh, but doing same job. And our Indian scientist in Hyderabad selling this Aditya in around 3, now I think there may be 3.5, but that time it was 2.9 or something, 3 lakh rupees. So Kavita was very much happy that now she can do the reaction. It's still working. Then few visitors has come. First batch of a student. Anyway, so those of you, this is my office actually, yeah. This is the purpose of showing this slide. My PhD supervisor, I must acknowledge him, because whatever thoughts process, he was very tough person from West Bengal. And I did my PhD from CDRI, Central Drug Research Institute, and he was like a taskmaster. We were always discuss the science, very tough guy, and but his toughness is very useful, become very useful for me in my today's life. So for the acknowledgement, therefore I am showing this particular slide. He was very happy, visited my laboratories, and there's a few master students, PhD students, and then these were CRSI conference has happened in JNU. There's Chemical Research Society of India. There's a society founded by CNR Rao in Bangalore, and so this conference was here. So I was the co-convener. And yeah, so this statue I hope you all have visited by now. How many of you from outside Delhi? अच्छा ज़्यादातर लोग हैं. So this is the Swami Vivekanand. His his statue is there near to the admin building, administrative building, and you must visit this place. And many people goes there, take the selfie. Then I thought, why not we should go as a group member? So we have taken the picture. So many of the actually now this Sunil is there. He has submitted the thesis. He has gone. He has submitted his thesis, and he is submitting the thesis. Relatively new batch, and then she was master student, gone to South Korea, now doing PhD in South Korea. And thank you very much for your kind attention. I hope I took little longer time than I have been given, and there was a purpose. Done. And I'm happy to answer. If you have any question, you write down the email also. There is an email, ram.sagar@jnu.ac.in. I am not on the Instagram or Twitter and all, but I use the email, and I have Facebook account also, but that was long back I have made, but ज़्यादा यूज़ नहीं करते, लेकिन है. मैं email पे मिलता हूँ, है ना, और phone पे.
Sir, can you share these slides?
Yes, why not? मैं slide ये कोई platform यहाँ बनाया है क्या जहाँ पे मैं upload कर सकता हूँ? व्हाट्सएप ग्रुप है, तो उसमें ऐड करना पड़ेगा ना. मुझे ऐड करेंगे तभी तो भेज सकते हैं.
नहीं, इनको सर YouTube पे मिल जाएगा, पर अभी सोशल मीडिया पे चला रहे हैं.
हाँ,
ये पूरा मिलेगा इनको लाइव.
मिलेगा. हमें भी मिलेगा.
आपको भी मिलेगा. आप तो सर ऑर्गेनाइजर पूरा आपके लिए.
So have nice stay in JNU, walk around and see the things, and get some bit of kind of transformed after 1 month. जब आए थे 1 महीने बाद अपने को देखिए आप कि क्या चेंजेज आए, ठीक है, नॉलेज के टर्म में, और भी आएंगे, तो वो आपका गेन होगा, एकेडमिक गेन, ठीक. एंड आई एम हैप्पी टू आंसर एनी क्वेश्चन है.
Is there any glyco-hybrid or drug that is in preclinical or clinical trials?
देयर आर मेनी. मैंने कुछ तो आपको दिखाया जो ऑलरेडी ड्रग में है, एंड देयर फ्यू मॉलिक्यूल्स आर इन क्लिनिकल ट्रायल आल्सो. तो कार्बोहाइड्रेट इज नॉट ओनली लाइक एनर्जी मॉलिक्यूल, इट हैज़ मच मोर एप्लीकेशन. अभी एक एस्पेक्ट जो टच नहीं किया, दैट इम्यूनोजेनिक रिस्पॉन्स, इम्यूनोजेनिसिटी, वैक्सीन, ठीक, उसमें भी बहुत ज़्यादा रोल है. इवन और ब्लड ग्रुप, किसका O होगा, किसका A होगा, किसका B होगा, कौन डिसाइड करता है, उसमें भी वहाँ पे और भी मॉलिक्यूल्स हैं. ग्लाइकन्स आल्सो प्ले रोल. Carbohydrates are molecular for life. Any other question? No? So thank you very much. I hope I imparted some of the new knowledge. Group photo?